In last video, we discussed that why the plant appears green in color and how the ATP and NADPH are energy rich molecules and provide energy to all the metabolic reactions. So, in this video, we will dissect the chloroplast and look into detail of every parts which is present inside the chloroplast. So, this is essential. Many of the questions have been asked from this part. So, remember that when we are discussing light reactions and dark reactions, we said that the location of each reaction is essential. Okay. So, keep that in your mind and listen this lecture. So, the chloroplast which is present inside the cell, each cell may contain many hundreds to thousands of chloroplasts in it, and each chloroplast is covered by a transparent membrane. And inside the chloroplast is a double membrane layer, which means that the outer membrane and inner membrane are there, and inside the inner membrane the portion which is known as stroma, and this stroma is nothing but which is highly like to the cytoplasm present in this cell. And there are another membranous structures are there. So, these membranes, this is unique membrane and we are giving many names to this. So, just listen carefully. So, if the membrane is separated or solitary, then we are calling that thylakoid membrane. So, you need to know that all the inner structure is made up of thylakoid membrane, and if it is solitary, then we are calling that predominantly in thylakoid membrane. Or in some region, the thylakoid membrane are stacked one upon the another, and it's look like we are arranging a coins or piling up the coins one upon the other. So, this structure is known as grana. Okay. And the membrane which is present in the grana, this is also thylakoid membrane, you need to know that, and this membrane we are specially given a name that granule membrane. So, the solitary membrane is known as thylakoid membrane. So, why I am very curious about this? Because the light reaction and the dark reaction both can happen in the granule membrane, and the light reaction which is little bit changed or little bit altered in the thylakoid membrane because of the lack of photosystems that we will discuss in a latest lectures. And inside the thylakoid membrane, there are many photosystems are there. The photosystems are nothing but which contains many chlorophyll and the accessory pigments. So, as we discussed in the previous lectures, the light is an electromagnetic spectrum and it contains energy. So, as the wavelength are smaller and smaller, the energy is higher. So, this you may know in that. So, this energy may disrupt the chloroplast, so that the accessory pigments are protecting the chloroplast by absorbing the excess amount of light or harmful radiations such as ultraviolet light or infrared radiation, so on and so forth. So, these pigments are given a special name according to its function, which is known as shield pigments, because it is shielding the chloroplast in order to protect it from the photobleaching. So, the photosystems contains many chloroplasts, many accessory pigments, and one reaction center chlorophyll. Okay. So, the reaction center chlorophyll is essential. This initiate the light reaction. That means, the light when it is hitting on the chloroplast and inside the thylakoid membrane and inside the photosystems, and inside the photosystem there are many pigments are there. When the light is hitting the photosystem pigments, the light energy is absorbed and the light energy is utilized for the excitation of electrons. So, when the electrons are excited, so the electron is always allowed to move in pairs. Okay. So, when the electrons are excited, then the excited electrons have much more potential energy, and this electron when it is moving from various protein to protein, the energy level of the proton, the energy level of the electron is getting lower and lower. So, at this stage the free energy is liberated. The free energy is utilized to transfer the proton from the stroma into the thylakoid membrane. So that the proton gradient is produced inside the thylakoid membrane or inside the grana. Okay. So, this electron, the pair of electron is still have a lot of energy, and this pair of electron now is entered into another type of photosystem, which I will give you the names of these photosystem little bit later. Then the next photosystem, when it has absorbed energy, this photosystem is also similar to the photosystem which we discussed earlier. So, the electron is now trapped into the photosystem, the second photosystem, and again when the light is absorbed, then the electron is excited. Now, the excited electron is directly carried out by the NAD plus. So, the NAD plus, when it is accepting the electrons, it converts into NADPH. So, now we know that the light hitting the photosystems, various types, two types of photosystem present in thylakoid membrane, and so that the pair of electron is excited. The excited electron have some potential energy. When the excited electron is moving to protein, it loses its kinetic energy, and this energy being utilized to transfer the protons from the stroma to the inner thylakoid membrane. And the electron reaches the next photosystem. Again it is excited, then captured by NAD plus and converted into NADPH. So, the first photosystem is named as photosystem 2, and the second photosystem we discussed in this lecture is named as photosystem 1. So, this naming is because this photosystem is observed or identified at first, then the later, so that the naming confusion is there. So, you need to know that the photosystem 2 first absorbs the energy and electron excited, then the excited electron is carrying on to travel from protein to protein, loses its energy, and it reaches to photosystem 1, then again excited, then again NADPH formation. Okay. So, now we have discussed the NADPH formation, and we need to discuss how the ATP is formed in the light reactions, and we need to know that the problems of these reactions. So, that we will discuss in the next